Hello everyone, I'm Sindoora Nalajala, a graduate student in the Institute of Plant Breeding, Genetics and Genomics at the University of Georgia, and I work with these amazing blueberries. Blueberry plants are attacked by a soil-borne pathogen called Phytophthora cinnamomi, which causes root rot and is one of the major concerns for blueberry growers. Identifying resistant plants can help improve blueberry production. Therefore, screening different blueberry plants is essential to understand their response to the disease. So in this video, I'll walk you through the steps involved in culture and inoculation of Phytophthora in blueberry. Let's start with the first step, preparation of V8 agar media plates. V8 agar media provides the nutrients needed for the growth of Phytophthora. To prepare this media, we weigh 15 grams of bacto agar, which is sufficient for 1 liter of media, and this serves as the primary solidifying agent in the media. Then we add 950 milliliters of distilled water into a measuring cylinder and make up the remaining volume with V8 juice. Next, we transfer the solution into a beaker and place it on a magnetic stirrer for even mixing. Once it is completely dissolved, we pour the solution into a glass bottle. The media is then sterilized using an autoclave. This is an important step because autoclaving eliminates unwanted microorganisms that could contaminate the media. After sterilization, the media is allowed to cool down to a suitable temperature. Next, in the sterile hood, we add 10 milliliters of antibiotic cocktail to this 1 liter of media. This cocktail contains 5 different antifungal and antimicrobial chemicals that prevent the growth of unwanted microorganisms in the media. We mix the media again on magnetic stirrer and the now media is ready to be poured into the plates. Carefully open each petri dish or plate and pour the media. In general, 1 liter of media can be poured into 35 petri dishes. And once the agar solidifies, the plates are ready to be packed and used for culture. Now, let's move to the next step, inoculation of Phytophthora on plates. In this step, we transfer Phytophthora pieces onto the prepared V8 agar plates. One of the easiest ways to transfer Phytophthora onto the new plates is by making agar plugs from a previous culture. For this, we use autoclaved pipette tips to cut through the media and form circular agar plugs. These agar plugs are then transferred onto a fresh media plate by carefully placing them upside down in the center of the plate using sterilized forceps. This helps ensure proper contact with the new media that is prepared. Moreover, this step should be done carefully using proper sterile materials to avoid further contamination. After inoculation, the plates are carefully wrapped with paraffin to reduce the chance of contamination and then kept in an inverted dark chamber to allow Phytophthora to grow. And this is how the culture starts growing in just 2 days. Once the culture has grown sufficiently, in about 7 to 10 days, it can be used for next step of inoculum preparation. The goal here is to expose blueberry plants to Phytophthora in a consistent and controlled way. For this, we use 4 plates of fully grown culture, peel off the culture using forceps and place it into 1 liter of water. Once the culture from all plates is added, we blend it with a hand mixer for about 1 minute. This breaks the pathogen into small pieces and creates an inoculum slurry that is easy to apply around the root zone. Once ready, we transfer the inoculum slurry into a bottle for the final step of inoculation of plants. The prepared inoculum is applied to the root zone. For this, gently remove the soil around the roots, then pour 50 ml of the prepared inoculum around the root zone, and finally cover the roots back with soil. Over the following weeks, the plants are monitored for symptoms such as reduced growth, wilting, and leaf discoloration to understand the response to Phytophthora and help in screening for resistance. Thank you for watching. I hope this video helped you understand the basic steps of culture and inoculation of Phytophthora.